Translation. So translation is the name of the process of making proteins from the genetic code. What we're going to look at first with translation is the cast of characters that are involved in the process of translating the genetic code into the protein. So the first of our cast of characters is the messenger RNA. So it's little m, big RNA. And the message that the RNA is bringing, or the messenger RNA is bringing, it's bringing a copy of the genetic code from the nucleus out to the ribosomes where the proteins will be assembled. So the messenger RNA acts as a blueprint. It's the instruction manual. It's a copy of the recipe. It's basically what your cells, what your tRNA, we're going to see, will follow in order to build your proteins according to the genetic code on the DNA, which is still trapped in the nucleus. So DNA, that double helix, it's just too big to leave the nucleus, but the RNA, that single-stranded structure, is small enough to leave the nucleus and go out to the ribosomes. So you can see the messenger RNA here in the picture to the right, that it will come out to the ribosome, and then its message will be read by the transfer RNA, and the amino acids will be linked together with peptide bonds to build your proteins. The next in our cast of characters is the tRNA, or the transfer RNA. Now, your transfer RNA is actually going to be transferring the raw materials to make proteins over to the ribosomes. So the transfer RNA, if you look at the larger picture on the left, it kind of sort of looks like a T. On one end of the transfer RNA is the amino acids. There's your raw materials. There's your monomers, your building blocks of proteins. On the other end, and there you go. On the other end is what's called an anticodon. So we're going to start looking at this genetic code as a sentence, a genetic sentence. And each genetic sentence is comprised of little three-letter words called codons. And that's the name of the code words on the mRNA. Their matches on the tRNA are called anticodons. They still follow the same rules using the AUCG as you would find in the messenger RNA. So the transfer RNA acts like the workers, bringing over the raw materials, bringing over the amino acids, and putting them together as it reads the messenger RNA as it moves through the ribosome. The last in our cast of characters is the ribosomal RNA, the little r, big RNA. And this is the genetic material. This is the type of RNA that's actually a part of the ribosome. Now, the ribosome acts like a construction site. It's a little protein factory. So all proteins are made here. And you can see the picture to the right. That most illustrations of ribosomes are a simple dot, and those are all completely incorrect. That there's actually two portions. There's this larger portion and then this smaller portion. And the smaller portion kind of connects with the larger portion to hold the messenger RNA in place, while the transfer RNA brings over the amino acids and puts them in place as they go, as it reads along the message on the messenger RNA. Now, these are linked together by little bonds called peptide bonds. So you can see that the end image of the protein, it kind of looks like a little necklace, a little beaded necklace. And what's holding these little beads, these little amino acids together, are called peptide bonds. So sometimes you might actually hear a protein being referred to as a polypeptide.